Dr. Wade Welshons, Associate Professor in the Biomedical Sciences at the Vet School at the University, and Carolyn from the Sierra Club. They've got a lot to tell you about the perils of plastic.
I'm giving preliminary information on Carolyn talk because I don't actually very well address sustainability. Mostly I detail the problems, and in some cases horrific problems, of not being able to address that. Now, I'm going to give a talk on chemical and environmental estrogens coming out of plastics. I'm part of the group here at the University of Missouri with Fred von Saal in biology and Susan Nagel in obstetrics, gynecology, and women's health at the medical school. And we've done some of the preliminary work, particularly on bisphenol A, and observations that it's much more estrogenic than was predicted by basic toxicology, and that those levels are in the human population, they're already in the current human exposures are already above the levels which in animals bring about effects, and which of course is very much too high. Classical toxicology would try to keep exposures to 1/100th or 1/1000th of the level which bring about adverse effects in animals. And unfortunately, this system has not worked very well for bisphenol A. And one of the, and I'll just get to what could be a question later, one of the reasons for that is that it's an environmental estrogen using polycarbonate, the lenses in my glasses. It has many favorable, desirable characteristics, but it's an enormous market. It's past $10 billion a year, which exceeds $1 million an hour, $25 million a day. That's an enormous amount of money to be able to buy mischief to derail appropriate toxicology. The handling of it in Europe has been substantially better by the standards of the science here, but it's still a problem, in part because of just the money involved. Oh, and the chemical industry hired the Weinberg Group about the time we published our first paper. These were the chief lobbyists for the largest tobacco company in the world, Philip Morris. So they brought directly to opposing findings or actions on bisphenol A by the use of experienced lobbyists. Sorry about my voice. It wavers because of an essential tremor. And my father had it in his hands, which would have been great because I don't do surgery anymore, but I got it in my voice. Sorry. Okay. Chemical and environmental estrogens in plastic, these matter because they act to either add to or block normal hormonal effects in normal physiology of the animal. And for chemical estrogens, there are a large number of synthetic chemicals which have the same activity or similar activity to the natural hormone. Well, this is the structure of the endogenous, the major circulating estrogen. It has a phenolic hydroxyl group. On the ring, it's a certain size. Actually, this group matters. It turns out that a hydroxyl group on a benzene ring, that's another shorthand, they're shorthand for these structures. A hydroxyl group on a benzene ring like that is a characteristic of virtually every single chemical estrogen there is, including all the endogenous ones. And yet, somewhat accidentally, this group, a hydroxyl group on a benzene ring, is very widely used in chemical, in organic chemistry and the development of pharmaceuticals and chemical products. And by, I think, what's essentially an accident, any number of chemicals are made which happen to be candidates to have hormonal activity, which is they can act similarly to to the natural estradiol, and by doing that interfere with normal regulation, because the animal body and ours constantly uses hormones, including estradiol, in normal physiology. So that's one of the reasons that estrogenic chemicals and environmental endocrine disruptors that are estrogenic are so important and so common. It's just, I think, an accident of modern synthetic chemistry. Now, the reason these molecules can be very active, that is, show activities, change development, normal effects at very low concentrations, is because they act as hormones, and hormones act by receptors. That is, there's a protein which binds the molecule, it's normally the normal hormone, but any molecule with the right shape, and that turns on the receptor, which goes on to turn on the response. So anything which can bind to the receptor in the right way will create an estrogenic response. And since estrogens are used in physiology all the time by the body, addition to or subtraction from normal function by an anti-estrogen or something which interferes with function will have to have a disrupting effect, and that's unfortunate. This is a space-filling molecule of what it actually looks like. The point here is just an illustration of this. What's diagrammed simply as a binding site for the receptor. An endocrine disruptor is here at the level of estrogenic activity, is a molecule which can bind to the receptor and typically activates it, although it doesn't have to. It can interfere with normal function, but typically it activates it. And the receptor, the bound receptor, is what brings about the response. And the affinity, the way that the chemical binds to the receptor determines how little you need in order to get a response. And that's what this arrow is leading to. Once it's occupied at one end, the rest of the molecule can turn on estrogenic activity or change estrogenic activity if this molecule isn't a perfect fit, or even block it. But since estrogens are used all the time in various functions other than reproduction, almost any interaction there changes normal biology and normal development. And one of the features to that receptor mechanism is that enormously small, that's backwards, extremely small amounts of chemical can be bound and turn on the response well below the level that can be detected by normal physical means. You can't see it. You can't see it with UV light. You can't see these concentrations with any but the most sensitive and expensive assays. And hormones and estrogenic chemicals are typically working at the range of 1 part per billion. That is 1 gram per billion grams, or 1 ounce, or 30 grams per billion ounces, or 1 drop in an Olympic-sized swimming pool of water, which is 10 to the something liters of water. The dilution is enormous, and it makes it a challenge to detect these by chemical methods, which also makes it difficult for toxicology to assess them. And current exposure to a number of estrogenic endocrine disrupting chemicals is right in the range to create an effect. It's not close to, it's above the range which creates effects. The one we looked at, somewhat by accident because it was just the most active in what we were looking at, is Bisphenol A, a molecule that is used beginning in the 50s, I think, polymerized to form polycarbonate. And polycarbonate plastic has a number of really favorable characteristics. It can be optical plastic. That's great for glasses. It adheres to metal, which a lot of plastics don't, and it's quite strong. So polycarbonate has been used more and more in bottles, and baby bottles are what we looked at, as well as the lining of cans to which it actually adheres, and bottles that don't break. In addition, dyes, UV blockers, various things can be added to it to make it softer or block light or add antioxidants. Many things can be incorporated into the plastic. But the parent plastic, the structure of it is polymerized bisphenol A, and when it breaks down, it releases that. And when the plastic is the plastic can be virtually completely dissolved. It's an enormous reservoir from the bottle onto its slope. Bisphenol A itself has come under study particularly for the last 20 years. And initially, in animal studies to pregnant animals, which create developmental effects, they change the development of the fetus in ways that are not reversible. That is, once the animal is born, the effects of the estrogenic activity then can't be reversed. And an example would be the effect of androgens on development to create the male body plan in fetal development. Once the animal or person is born, taking away the testosterone won't reverse the development. The male body plan is already in place. Well, that applies to many other kinds of physiological effects because estrogens have so many in development. And some were somewhat surprising over the last 10 years or so. At any rate, in animals, early exposures created permanent changes in the subsequent adults involving obesity, increased animal embryo mortality, changed breast development and larger prostate actually mean now precancerous changes in the epithelium of those organs. Now, prostate cancer, changes in the prostate in experimental animals don't lead to prostate cancer as often because it takes so long to development in people like us. But it does create the precancerous changes. In addition, surprisingly, earlier puberty, lower androgens, they're developmentally animals are somewhat estrogenized, and then altered estrocycling. This is a curiosity in rodents. They have 3 or 5 day estrocycles, or certain strains do. When they're estrogenized, they all have 5-day cycles. If they're not, they're 4-day cycles. They're also more fertile. They cycle longer at any rate. Exposures to the animals have created a number of effects as well. These are experimental exposures, including increased insulin resistance, changes to the immune system, and hyperactivity or aggressive behavior. And each one of these here is associated with current human epidemiological concerns and trends. Now, I'm not saying they're all caused by bisphenol A. I mean, I'm old enough that I've seen cycles of these, and it's almost never what it seems like at first, but it can lead to what the real problem is. But certainly at a superficial level, most of the current epidemiological concerns can be brought about by exposures to bisphenol A to animals and people at current circulating levels, which is unfortunate. Now, epidemiological studies mean studies where populations have been studied with some kind of measure of exposure and some kind of association with an effect. That association is what's termed here, because it's not cause and effect. Because you can't ethically do the experiment where you take 2 groups of people and give one group something you expect to harm them. You can't do that. Clinical studies usually involve best current treatment plus something to see if it improves it. The human effects will always be associations unless they're accidental. But epidemiological effects have associated increased exposure to bisphenol A as circulating or excreted material to things including earlier puberty, cardiovascular disease. This was completely unexpected till 10 or 12 years ago. And then other kinds of problems, reproductive problems as well as problems that aren't related to it, diabetes, type 2 insulin resistance, and so forth. So the point is that bisphenol A exposure, this environmental estrogen, which has been studied extensively, shows a number of associations in humans with just the kind of things they produce in animals, which is about the level at which cause and effect is inferred. That is, people start avoiding it, even though there's no technical proof. There was a review of about 75 studies. I listed this here because it includes asthma, of all things, probably an immune problem. That is, associations of higher exposure to bisphenol A to the mother of the children and subsequent asthma, as well, of course, as behavioral changes. I mean, it's really surprising in a very negative way. Well, in 2006, we were asked by Environment California. Now, this is when polycarbonate baby bottles were in use. We were asked to look at how much or to what extent bisphenol A was coming out of polycarbonate bottles, that is, degrading to release this estrogenic chemical. Well, Environment California, determined what the 5 in their area, in Northern California in particular, their area which were the most commonly used, the most popular polycarbonate baby bottles. And then they got samples of them in different stores, so you couldn't say one store was selling something that was hazardous compared to another. And then we extracted them with water under conditions that familiar with for similar to the dishwasher and the sanitize cycle. Anyway, these are the 4. And we compared them to glass baby bottles as something which is not polycarbonate. And I know to half the audience probably this is extremely boring and uninteresting, but just let me point out that, in fact, from all the bottles, we measured BPA coming out of it. The total amount per bottle, on average, was around this number, 2 micrograms per bottle. So just keep that number in mind. 2 micrograms per bottle. And for baby bottles to infants, that is in animal studies, that's a biologically significant dose. It's in the range in animals that creates effects. Now, Fred's lab, Dr. Julia Taylor and Fred's lab measured the actual bisphenol A coming out. And my lab, Dr. John Flanders, he was a vet student then, he measured total estrogenic activity. And the question was, is there something else coming out besides bisphenol A? And statistically, these matched up. The estrogenic activity expressed as the amount of bisphenol A it would take to make it that estrogenic. Doesn't mean that's what it is. That activity was about the same. So it didn't look like there was a surplus, except one of the bottles. And John eventually determined that that was due to the closures. You know, the nipple, the disc that can be used to seal a baby bottle, even though it's glass. So the estrogenic activities were coming out of that. And, well, okay, and this in California, well, Canada, Environment Canada, then got polycarbonate baby bottles banned in Canada. That transferred here, and for a month we were unable to get glass baby bottles for our own studies because they were completely sold out. But eventually, or pretty quickly, BPA, bisphenol A free bottle replacements ended up on the market. They just flooded the market. So, great. Well, the thing is, there's an enormous amount of plastic, and this is from Pete. He took this from Flickr. It's a canal, I think somewhere in Asia, Thailand, Bangkok. I don't recognize the picture. But this is the plastic in canal. It's an enormous amount of plastic. And one of the questions was, are the bisphenol A-free bottles really BPA-free, and are they estrogenic themselves? Do they have something else in them? Well, a group in Texas at Certichem, a company that measures estrogenic activity from anything, they reported that they tested many plastic products, including bottles, that had this very extreme method of testing them. They essentially dissolved them when they could. And they tested plastics that were not polycarbonate, but just about everything else. And they found estrogenic activity in a large fraction of them. I didn't believe that for a minute. I knew these people, and they didn't run, at any rate, experimentally, there were controls missing that looked suspicious. So we collected bottles and we took a look at some BPA-free bottles available in town. And what we did with a given bottle, we didn't use very many. I expected to see nothing. I was wrong. It was about 80% of the time. I expected nothing, so we did the experiment to be very simple, very minimal handling, so we couldn't contaminate, just minimal technique. And we took a bottle, and we checked a new bottle, and then we autoclaved it, that's for heat. And then we checked it again. And then we microwaved for radiation to a limited extent. And then we gave it 24 hours of UV exposure, long UV, that's, strong, heavy, extensive sun exposure. I mean, we were modeling these things, and the prior paper did that from Certichem. And we then, these little profiles here indicate measures of activity, estrogenic activity, and the glass bottles we had with every treatment, so this is one bottle treated over and over by one method or another, they were completely flat, and that's why they're green. No estrogenic activity, no toxicity, as it turns out. Well, these two bottles here, this is two of a set, first, they all showed something coming out of them no matter what we did. We washed them 3 times by hand, autoclaved, microwaved, UV, burned them, and no matter what we did, stuff just kept coming out. And it wasn't normal estrogenic activity, it was a combination, which makes sense if you think of toxicity. You get chemicals that may be estrogenic, environmental estrogens, as well as things which kill cells. Oh, and these studies were in human breast cancer cells and human endometrial carcinoma cells, so they have clear human relevance. They make these cells grow faster, and these cells have been extensively used models of human hormone-dependent breast cancer. So the effects we see here would be expected to show the same kinds of effects on human breast cancers. At any rate, the surprising thing was one, there was tons of what I'll show you in a minute. There was a lot of activity, and it just didn't go away. None of these treatments got rid of it. It was very resilient. And side and controls, glass right next to it, same treatments, same washing, same everything, showed nothing at all. This is the whole panel. These are BPA-free plastics here, 7 of them, and then 2 columns of glass bottles. And we have a number more, but this is about what one can see. And the thing that was just very surprising to us was that every single bottle, each bottle, and every treatment extracted substantial estrogenic activity in chemicals. And some were purely estrogenic, like polycarbonate. 2, with whatever the treatment was, were purely toxic, but most of them showed both activities, which was not expected. Well, we can use that assay to measure how much estrogenic activity is coming out of these bottles as bisphenol A equivalents. How much BPA would we have to add to get that much estrogenic activity? So it's kind of a backwards assay. Well, what we found here, these are individual bottles. These are the treatments going from a new bottle down to one that's gone through and UV radiated all the way through. These are the bisphenol A equivalents of estrogenic activity. None of them had very much BPA. Well, these are the polycarbonate bottles in terms of estrogenic activity. And these are the bottles marketed to replace the polycarbonate. They're 10 to 100 to 1,000 times worse than the bottles they replace in terms of estrogenic activity. Well, now these are preliminary data. They're not published yet. We aim to have that done next year. Certain person or two in this room will help with that. So I can't give you the bottles yet, but these are just things bought in town or on Amazon. These are ordinary water bottles. So BPA-free bottles in ordinary use appear to release a lot more activity, estrogenic hormonal activity, than the bottles they replaced. And this continued through stressing. That is nothing we did about it. But now we, in a normal lab setting and time scale, this was out to 2 or 3 years. The plastic in the oceans can be there for much longer, but certainly our indication is that plastics in landfills and circulating in the oceans can be releasing things. Some of them can be releasing all kinds of things for a very long time. And that leads me to this, which is Carolyn's slide of the Great Pacific Garbage Patch. Our inference, without testing it, would be that some of these things are releasing endocrine active, endocrine disrupting chemicals for a long time.